Okay, class. We're going to call this lesson 17, which is going to be PowerPoints from the lecture 14 PowerPoint file and Canvas. And this PowerPoint covers population genetics, which is a whole chapter in your text. Okay, so let's talk about population genetics. If we're going to talk about population genetics, we need to define a population. We're going to call a population a collection of interbreeding, which means that they're breeding and they have viable offspring, not like Napoleon Dynamite's favorite animal, the liger. It has to be able to have offspring of a species, which is the definition of a species. So that's a population. Polymorphism, we talked about this word. Poly means many, morph means type or change. So polymorphic is different variations in a population, and then we're talking about the DNA level. And so this is at the alleles, right? Because not normal. Most populations are diploid, so they have two copies of alleles for every gene. But there could be more. I mean, we talked about strawberries. They have 8 copies. They're octoploid. But we're just going to focus on two because when we throw in a bunch more like triploid and tetraploid, it gets a little complicated. So that's what polymorphism means. And so we talked about this before. We talked about mutations and stuff. But I just want to remind you that when we're talking about alleles, uh, it, it the difference in allele can come down to simply the DNA sequence. It doesn't mean that it has to change the function. It doesn't mean that we have to see a different phenotype. It can be any change, and that change can be anywhere. And we talked about this. It could be in between uh, genes. It could be in introns, intergenic spacers. Uh, it could be anything. It doesn't necessarily have to change the phenotype. So any change can be a polymorphism. All right. So when we talk about monomorphic, that means that everything's the same. And we kind of discussed this, right? We're not really interested in this. Just like, we talked about this. You wouldn't describe the thief as having black hair if every human being had black hair. So we're looking for variations here, and so that means they're polymorphic. So more than one change. This is showing you an example. We have these individuals with a G here. This individual has an A, G, A, G, G, A, and so on. So there, in this population of individuals, there's either a G or an A, and that's a polymorphism. And we're interested in that. And then here, there's either a T or a C, right? And so that's a polymorphism. The rest of the stuff isn't changing, so we don't really care about it. And that's how we were looking at sequencing, doing those single nucleotide polymorphisms, or SNPs. Oh, I gotta look at my thing. So this area right here, since it involves a single nucleotide, and same thing here, these are called single nucleotide polymorphisms, which we've talked about. And that's how, uh, 23andMe and Ancestry or whatever, they're not sequencing all this other stuff. They don't really care because it doesn't vary in a population. They're focusing on these individual uh, variable places in a population, and there's roughly 1 million of those. And it's a lot cheaper to look at 1 million spots than it is to look at 3.14 billion. All right, that's a order of magnitude like several thousand fold greater. Okay, so if we look at a variation in a population, and you know this, it's not a surprise. if you sent your DNA off to Ancestry or 23andMe and they looked at all of your SNPs, your single nucleotide changes, and they compared them to other people, you know that your DNA is going to be most closely similar to someone that's maybe your sibling or your mother and father or your children than it is to someone who's, lives in another country. And so you know that the more closely related you are to an individual, the less variation you see in your DNA. And that's basically the way that Ancestry is able to tell you who's your second cousin, who your mom and dad are. And my wife did this and found she had a long lost aunt that her grandfather never spoke of. So, anyway, the DNA doesn't lie, it knows. And when you have if you're trying to identify a person using a million markers, like what color's their pants, do they have a stain, do they have a tattoo, how tall are they, then chances are you're going to narrow that down to like one person. So if we look at variation in humans and primates, right, monkeys. Okay. So, sorry, I had someone come in my office. So if we have two homologous sequences from unrelated human individuals, so we're going to compare two. Remember, homologs are similar but not the same. So we're going to compare one allele or one chromosome from another, some sequence. It doesn't even have to be a gene, it could be anything. Um, if we look at that, about 0.1% of all the nucleotides would be different, right? One, like, so 0.001, 1/1000, right? And so we kind of talked about that. Like, there's a million variations. There is about 3 billion. So that's 1,000 orders of magnitude variation. So we're just going to look at those SNPs, and we would find that we have about a million variations out of our 3 billion uh, nucleotides that make up our entire genome. If we looked at sequences from unrelated, so we're just randomly picking a chimpanzee out of a population, they're not like cousins or closer, about 1.2% of their nucleotides would be different. So what does that tell you? Well, if we know that we're looking at a family tree, and the further back that tree is, the further away from you, let's say this is you, the further away from you is going to be more variation, right? And the closer to you, like mom and dad, is going to be less variation. And this would be, more or most variation. And so, what does that tell us about, well, chimpanzees and humans? And what it tells us is that we, as a human population, have less variation than chimpanzees do in their population. So, which one's the newest population? And that would tell you, well, it would make sense that humans are, because humans have less variation in their DNA, 0.1%, as opposed to chimpanzees that have more variation, which is 1.2%. And we can use this to try to rebuild a tree, not unlike a family tree. Now, if we looked at differences between humans and chimpanzees, we would see that about 1.8% of the nucleotides are different. So we could look at the tree and say, okay, these are humans, these are other related humans, and, chimpanzees maybe would be over here. So how do we know this? Well, we look at SNPs, we look at the single changes in DNA, we look at the microsatellites that we talked about, right? Those small repetitive elements, like GATT or GATA, and then they repeat GATA. So there's trinucleotide, tetranucleotide, dinucleotide repeats. And then we look at insertions and deletions. We can also look at mitochondrial DNA. Sometimes this is easier because if we compare mitochondrial DNA, we don't have to worry about it recombining. We don't have to worry about crossing over. We don't have to worry about any of that stuff because in most, I wouldn't say all, but a lot of organisms, the mitochondria only comes from the mother, which we talked about. And then chloroplasts are the same way. So we can look at mitochondrial DNA, which, is generally was done initially because that's easier to say, okay, this person, mother has the same mitochondrial DNA as this person, so they have to be the mothers. And then we also looked at Y chromosomes because those don't vary. Remember, there's not any crossing over in Y because only the ends are homologous. So mitochondrial DNA, Y chromosome is very easy to look at. Chloroplast for plants is very easy to look at. And then we can look at chromosomal variations. But when we do that, we have to take into account that people are going to recombine their DNA. So, for example, your mother and your father's DNA came together to make you. Okay, and then we can align these. So there's lots of programs that can do this, and this is done with a computer program. Just to let you know, there's a sector of what's called bioinformatics, and I have a lot of students that went into this. So basically, it's using computers to gain information about biology. And so someone would write a computer code that would align these sequences. I mean, remember, this genomic data is huge. 3 billion letters. I mean, I think we may have talked about this, but that would be the equivalent of a textbook the size of the Washington Monument. That is a lot of data to go through and analyze. And so we need computers to help us do this. We would never figure this out on our own. So we're just lining up sequences between different individuals. We have 7 individuals. These are each of the nucleotide positions that we're looking at, 1 through 35. And then we can classify them by haplotype. What's their type? And then we can, do a haplotype class to group those as well. So just looking at these sequences, we can see that these are all the same, right? This is all the same. There's a C variation here. There's no variation at this site at all, or here. So we don't care about that. There is a variation at this site between, half of the individuals. There's only one change in this individual, that person, number 2. This is a insertion or deletion. Probably a deletion. Those are more common. Again, we'd have another insertion or deletion here. Here's some microsatellite data just to show you. So let's see if we can find the repeat. AG, 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 AG. So I'd say the repeat is probably a dinucleotide repeat. And it's this AG here. So, and then some individuals, remember we talked about this, they only have maybe 3 repeats where these individuals have 5. And that's how we can identify one person from another. Here's a single nucleotide polymorphism in one individual. These are all the same, so that doesn't affect anything. And then by going through all this data and looking at how closely each sequences to one another, and there's other stuff that you can look at, like, when we talked about the genetic code. So the 3rd position of a protein coding gene is mutation has less likely to have an effect as the 1st position. We look at the genetic code, at the end of the sequence, it could be an A, a G, a C, or T, and those might all code for the same amino acid, which wouldn't change the protein at all. So there are other nuances to this that you can write into the programs and have, maybe this not count as much because it's free to mutate more quickly than other sites. But anyway, there's all kinds of analysis you can do, and then you'll have a computer program that was going to define these groups of how they're related to one another by developing what we call a haplotype network. All right. So how do we know the age of the human population? Well, we can do that using a technique called molecular clocking, and I'm going to explain it to you. But essentially, just think about it this way, and this is a very simple, dumbed down explanation of it. But if mutation occurred at a certain frequency, let's say that every day you got one new mutation. Well, if you had 365 mutations, I would know that you've been around for a year. And if you had 3,650 mutations, I could say you've been around for 10 years, and so on. Now, there's going to be some variation here. You might get to the same mutations in the same spot. Sometimes mutations are deleterious, so they would kill you, uh, or they may be advantageous, so we kind of look at that. But, on average, this is what's going to happen. And it helps to look at a whole population. It's kind of like if you're doing one coin toss and it comes up heads, the chances are that it might come up tails the next time, but it's not always going to come up heads. And the more you flip the coin, so we flip it 10 times, and you can do a chi-square on this if you want. We flip it 10 times, right? It's more likely to come out 5 out of 5, but you get a lot more variation in this. So 6 out of 5 or even 7 out of 5 wouldn't be unusual. But if you flipped it 100 times and 70 out of 100 occurred, that's going to be way above the chi-square value. So this number is going to matter the most. So if we look at more individuals, then this number is going to be closer. Just like Mendel, right? The more peas he counted, the more plants he counted, the closer he got to that 3-to-1 ratio. So anyway, if we look at the whole human population, we can use this data, and again, this is a dumbed down version of it. There's a lot more sophisticated things going on here, but and I'm going to explain it. But that's called molecular clocking, right? We're using the amount of DNA variation that occurs over time to try, and then we calibrate it against the fossil record and other things that we know, and we can get a good idea of where a population started or how old it is. So, a graduate student named Rebecca Kahn, which I know well, went to the University of Hawaii, which I was super jealous of. She studied DNA, and she studied DNA from the mitochondria of females. And what she was looking at is how much that varied. Right? So it was quite the interesting project. And when she was all said and done, she could figure out that all of the individuals that came from one lineage, right? All humans, all female humans came from a single lineage. So there wasn't enough variation in that to show that it came from another lineage or whatever. There weren't two groups. And then she could use this data, right? How much things vary over time to clock this. And she came up with a number of about 150,000 to 300,000, somewhere in this neighborhood. And there's some error in here. Years old. So years old, years ago. Um, and so okay, so that was interesting. So and she was able to figure out, like, just like if you had a bunch of family members um, that were their tree was branching off like this. If you looked at all the family members and you went backwards, you would be able to see what great grandmother they all had in common, right? So you're basically reverse treeing this. And so what she found out is where that individual came from, and that was Africa, and about that time period. So this guy named Michael Nachman, who is, I know him as well. I went to East Carolina to do my master's, and he was applying for a position there. He ultimately ended up taking a position at University of Arizona. I'm not sure if he's still there, but I'm pretty sure he might be because he was tenured. But anyway, he looked at Rebecca's data and said, I'm going to double check this. And so he used the Y chromosome to do the exact same thing she did with the mitochondria. And I put these papers in Blackboard, by the way. And what he found was that his data fit very well with Rebecca's data. It said about, 200,000 years ago in Africa was the original male. Now, does that mean that we came from a single male and a female in Africa? Maybe not. Probably not. Nobody knows unless they make a time machine. But um, it could be a population of individuals, right? And maybe something happened. Like maybe there were humans before, so there were lots of humans before, and something happened, and they all of them died. It could be a disease like HIV or the Black Plague, whatever. Some of them had mutations that allowed them to survive, and then they repopulated the Earth, right? So we call that a bottleneck, and we're going to talk about that as well. So anyway, and I'll tell you a personal story. Like molecular clocking is pretty accurate. Uh, I was told I did my master's degree on the relationship between all the land tortoises. So there's tortoises in Africa. You've probably seen them run around. They're popular as pets. There's actually several different tortoises in Africa. There's tortoises called Unifera. That's the species name. Giacalone is the genus. In Madagascar, there's not many. There's only like four in the U.S. and they're protected and they're on an island off the coast of Georgia. Uh, there's not many in Madagascar, so they're almost extinct. And we'll talk about why that's a problem. There are other tortoises in Africa. There's a leopard tortoise that kind of has leopard spots, and there's more tortoises in South America. um, there's one in Chile. There's a redfoot, a yellowfoot, and so on. So I looked at all these in the Galapagos Island, and there's giant tortoises in the Seychelles as well. And that's another story that kind of an interesting story I'll tell you after I finish this one. But so anyway, I was told I did this study and I looked at the tortoises at the Galapagos Islands and I did the molecular clock, and the molecular clock told me that those tortoises diverged from a population called Giacalone chilensis from Chile. They probably floated to the Galapagos Islands and set up a colony there. Um, and so that's what my data said. It was 3 million years old. But if you look at the fossil record at the time, and this was, in the late 80s, uh, they said that the Galapagos Islands were only 1 million years old, right? So this data was way different. And in fact, I had really a crisis of conscience. And I went back and checked my data, checked the DNA, resequenced a bunch of the tortoises, went back, triple checked it, quadruple checked it, and I kept coming up with the same answer no matter what I did. And so I was pretty confident in this value. So I published that paper, and all the geologists had a complete meltdown over it. In fact, I got heckled at the National Ichthyologist and Herpetological Conference in New Orleans, where I presented this paper in a public setting. And some people got up and said, this is bullshit, and walked away. So as a graduate student, that's pretty crazily demoralizing. But I stuck to my guns, and about 5 years later, they found out that this number was wrong. That the islands had risen and fallen over the years over sea level, and so the older ones were underwater that they never checked. And when they've looked at the fossil evidence and did a bunch of other studies on this to determine the time, they came up with the same number, which is pretty crazy, right? Even though I was told that I was 100% wrong. This just goes to show you cherish your exceptions because you might discover something new. Um, so, anyway, molecular clocking is quite accurate. I mean, back in the day, when you looked at like DNA comparison and they, had a DNA from OJ at the crime scene, uh, most people didn't know what DNA was or didn't know the power of it. I bet if someone today said, "Hey, your DNA matches mine, 25%," you'd be like, "Oh yeah, well we must be related," and you wouldn't question that. Um, so it's come a long way, but molecular clocking and this technique is very powerful to look at populations, how they're related, and how old they are. All right, so I just kind of threw this in. If you're interested in doing this stuff, or this interests you, it's a pretty good gig to do bioinformatics. And there are some programs that have been written. This one is written by Sudhir Kumar at ASU. There's another one. So there's lots of these sequence alignments and software that can generate these relatedness trees. I'm not going to teach you that. That's for a bioinformatics class. But if you're into that or you're interested in this at all, then I strongly suggest that you take bioinformatics. It's going to probably be pretty invaluable relatively soon when people start generating their own DNA sequences, and then medicine goes in that direction to make personalized medicine. Okay, so and then I promised you the other story. So when I was looking at, I didn't go to the Galapagos Islands. I went to zoos and I got blood from tortoises and isolated their DNA and did PCR and sequenced it. And what was interesting is I had all these different tortoises and these different populations, but this one tortoise from the Chicago Zoo kept coming up really weird in my tree. And I was like, is that my DNA? Did I contaminate the sample? What's going on? And I went back to the records in the Chicago Zoo, and that tortoise was extinct in the wild. And it was actually collected by Charles Darwin in the early 1800s. So that thing was like 100 and 50 years old or something, like 160 years old, uh, whenever I took its blood. That was pretty crazy. I didn't really expect that. But again, I wouldn't have known that if the DNA data, the alignment, didn't tell me something was weird going on with that turtle. Anyway, all right, so we know this. This is not a surprise that not all regions of DNA mute at the same rates. You're going to have more mutation in introns than exons. Makes sense, right? Because introns are thrown away. And they don't really matter. The only thing that matters is where that spliceosome binds. So those couple of sequences that we talked about, that they have to exactly line up the spliceosome. But other than that, other than the intron exon junctions, it doesn't really matter. It's going to mutate faster in non-critical promoter regions. So we talked about this. The space between the -10 and -35 doesn't really matter. So those sequences are going to be more free to mutate and not cause an issue, as opposed to being right into the -10 or -35 region. And I showed you that slide that really cuts that down. And then we have areas between genes. They don't really matter. I mean, we are finding that some of these intergenic regions do have an effect on phenotype, but it's pretty rare, and so they mutate faster. Um, even if it isn't a region that codes for a gene, then we talked about this, we can have different kinds of mutations. Some mutations accumulate faster than others, so like the third position, um, and it can depend on how important that gene might be, right? So if you were going to look at something like hemoglobin versus hair color, what do you think is going to be more conserved? And hopefully you answered that hemoglobin would be, because if without that you would die. Your hair color doesn't really matter uh, in humans because we're not really being predated on. No one's There's not mosquitoes that are picking out blonde people and giving them malaria. All right. So we can classify the mutations that occur by how it affects our fitness. Um, and so this is the classification, and again, this is just how geneticists talk. So you're gonna need to know these terms. So write them down, make a flashcard, whatever you want. Um, so the mutations we're going to talk about are called neutral mutations. That means that that mutation doesn't change the fitness of the individual. You're not less fit if you have blonde hair versus brown hair. Um, you're not less fit if you have a longer little toe than if you have a shorter one. So those kind of mutations, I call whatever, it might not affect your fitness. Well, I'm not going to get into specifics, but eye color could affect your fitness, and so that's why there's variation in eye color. But there are things that don't affect your fitness, and we talked about those. So there are mutations that are bad, right? They're bad for you. They do change your fitness, and they do it in a negative way. So they're going to decrease your fitness. Um, an example of this might be sickle cell anemia, right? So if that mutation occurs, that would it decrease the fitness in individuals? They won't be able to run as far. They might not be able to escape that cheetah chasing them or whatever. Their children have a higher chance of being homozygous. Uh, there's all kinds of like things that would affect your fitness uh, from that mutation. Uh, and remember, fitness really depends on if you were able to pass that on or not. So if you die before you're able to have offspring, it might not affect your fitness, or it would if you died before you could have offspring. It would affect your fitness more than if whatever that mutation was allowed you to reach reproductive age and have offspring. Um, and we'll talk about that in a minute. And then, of course, there's beneficial mutation. So, probably like every probably boy, I don't know, kids dream about superheroes. So, if you develop the ability to shoot lasers out of your eyes, that might help, right? And if you could fly, that might help you out too. So there's mutations. So one of the mutations that this is a real one, not lasers or whatever, would be your ability to break down the sugar in milk. So your ability to use lactose as an energy source would be a beneficial mutation. And we talked about this. So fitness is reproductive success, right? Your fitness changes. It's worse if you. I mean, it doesn't really matter as long as you can reach an age old enough to have children, and then it's not going to affect you. Let's see, what is this caution that I put on here? Right, so environment could be important too. We'll talk about that, but um, so peppered moss, right, are a good example. They come in two flavors. They're either black or white, and so they lived on these trees that had white bark, and that was before the Industrial Revolution. So birds would fly around and look at for moss, and so what do you think stood out more on the white tree? The black moth or the white moth? The answer is the black moth. So the black moss That would be a deleterious trait, allele, mutation to be black in that environment. And then they built all these factories around these trees and they started turning out smoke and smog and whatever, and it turned the tree bark black. So in the end, uh, the black trees, what do you think the birds picked off first, the white moss or the black moss? And the answer is the white moss. So that's a great example of how an environment can affect your fitness. Um, all right. So neutral mutations. Most sequences, uh, the polymorphism that we see are neutral. Most mutations are neutral. They don't affect your fitness. Um, they don't really affect much of anything. Um, that's because these changes occur in things that don't really have much to do with biological function, like we talked about introns, intergenic regions. We talked about this. So the intron edge-on junctions are important. We can have synonymous changes, so it doesn't change the amino acid. We can have some synonymous changes that have little change because it doesn't change the side chains, so they're chemically similar. So we've talked about this. Any of these things would lead to a neutral mutation. There's no benefit or harm in those particular mutations, and those are some of the reasons why. So we've talked about that, and we've talked about this, right? So we talked about how some amino acids are positive and negative. So if you had a positive one replaced with another positive one, it wouldn't be that much of a big deal. We can have ones that are non-polar versus polar. So there's amino acids have different. I'm not asking you to memorize the amino acids or what their uh, chemistry is, um, except for to know that it has an amino and a carboxyl group, and that affects the shape of it. But you don't need to know individual amino acids. All right. We talked about variable number of tandem repeats. You know that when we have these microsatellites, we can have 5 repeats or 2 repeats. So that's not. I'm not going to spend a lot of time on that. We can look at populations, right? We can use this variation to identify one individual from another, and I showed you the video on this. This would be an example of the class size. This would be repeats of 3 or 4, 4 to 5, and so on and so forth, and then greater than 14. So in this population, we have individuals, 11 individuals that have 9 to 10 repeats for at this locus, so D14S1. That's just a region of DNA, right? It has a crazy name, but you know, it's just what it was named, like because it's not in a gene. So it's D14S1. And so this, if we looked at this region, we would see repetitive sequences in here. I'm not sure exactly what the repetitive sequences in this. Actually, let me look it up. So I wasn't able to really find it. All I could find is that it's a RFLP region and that it's on chromosome 14. But anyway, it doesn't matter. We could just say it's AT or whatever. It could be a trinucleotide repeat. But there's this number of repeats. So 105 individuals in this population, and 3 have 1 to 9 repeats, whereas you get a like much broader range of this. And then if we look at another locus, so here's another locus, D114S13, we can look at the variation in that as well. So 108 individuals, much more variable at this locus. Uh, this group, this tribe group, the Saruri, have very similar variation in this locus. So a lot of variation. All right. The reason I'm showing you that is that it's a polymorphism and we can use it to identify or look at relatedness between individuals, which we've already talked about. Okay, so let's talk about Hardy Weinberg. Can't do population genetics without those guys. So Hardy and Weinberg were at a bar drinking and talking about they're population geneticists, and they're talking about how to calculate the frequency of things in a population without knowing it. Right. So let's go back to purple flowers. So if I said okay, there's a field, and in that field we have 70 purple flowers and we have 30 white flowers. And I say, go out there and pick me a purple flower. So you bring it to me. It's purple. And then I say, what's the genotype? And the correct response would be, I have no idea, because it could be big P, or big P, little p. The only one I know about for 100% certainty what the genotype is white, because I know white has to be little p, little p. And so basically what they were trying to figure out is there a statistical way to figure out the probability that that purple flower or anything in that population would be heterozygous or homozygous? And they came up with the equation, which is, I mean, pretty simple, actually. They did it on a napkin at the bar. So they said that the frequency of allele 1 was called, we'll just keep with big P, plus the frequency of allele 2 squared equals 1. All right, so you guys have done FOIL before. So if we did this, we'd have P plus big P plus little p and big P plus little p equals 1, and we multiply those out. We'd get P squared. Then we'd have big P little p for here, and then this one, the other way, would be little p big P. They're the same. I'm just going to flip them around because it doesn't matter the order of the allele. And then the last one would be little p, so that would be little p squared. And because all these p's are confusing, they said, okay, let's forget about that. Let's call this P and Q. Right? So Q represents the recessive allele and P represents the dominant allele. So anyway, if we finished out this equation, we get big P squared plus 2P, big P little p, plus little p squared equals 1. Right? Because these p's are confusing, they changed it to P and Q, and so they said P plus Q squared equals 1. Same thing. We did it out. P plus Q times P plus Q equals P squared plus 2PQ plus Q squared. Those all equal 1. Now, in this case, it's the same thing. P squares represents big P, big P. In this case, it's homozygous dominant. This represents the heterozygotes, right? And there's two of them. So that's why we have 2PQ. These are heterozygotes. And then this last one is homozygous recessive, because that represents little p squared, right? And so Hardy-Weinberg's equation says that homozygous dominant frequency, right, percent in the population, plus the 2PQ, the heterozygotes percent in the population, plus the homozygous recessive percent in the population, all those add together have to equal 100%, or this is 1, right? Because a percent would be a decimal. Like 0.5 is 50%, in case you didn't know that. So anyway, that's basically what Hardy-Weinberg did. And so they figured out that they can figure out what the probability of the frequency of this is by only knowing this value, right? If we have Q squared, which represents homozygous recessive, we can figure out all the other ones. And this is how it works. So let's say that let's go back to our population of flowers. And we said that we have 100 flowers and 70 of those are purple and 30 of those are white. And so that means that if we do p squared plus 2pq plus q squared equals 1, well, we know that q squared is this population here because that's the only one we know. We know that that has to be homozygous recessive. So we know that q squared equals 30, right? The part divided by the whole, which is 0.3. And if we square root q squared, that will give us q. So q equals the square root of 0.3, which is 0.55. It's 5477. I'm just rounding that. So if q equals 0.55, then p must equal 1 minus q because the frequency of a and of big A and little a, if big A is 40%, then little a has to be 60% because together, if we add them together, they have to equal 100%. So we know that P plus Q must equal 1. So if we just rearrange this equation, it tells us that P equals 1 minus Q. So P is going to equal 1 minus 0.55, which is going to be 0.45. And now we can calculate 2PQ because we have P and Q. So, and we can also calculate P squared. So we have 2 times P, which is 0.45, times Q, which is 0.55. And that's going to give us the percent of heterozygous. So 2 times. So that gives us 0. I'm just going to call it 5. It's 49.5%, but I'm just going to call it 50%. So we would expect 50% of that population of flowers to be heterozygous. So half of all of these would be big P, little p. And then what are the ones that would be homozygous dominant? Because we can't tell the difference. And so we would have p squared, which is 0.45 times 0.45. And that gives me roughly, I'm just going to say it's zero point 0.2. It's really 0.202. That rounds down, so we're going to call it 0.2. And then our original amount was 0.3. So if we go back to check our work, p squared plus 2 pq plus q squared, well, we calculated p squared to be 0.2. We calculated 2pq to be 0.5. And we also calculated q squared to be 0.3. And so all of those should add up to 1, right? That's the equation, and they do. So that's a good way to check your work. And that's how to do the Hardy-Weinberg equation. So in a diploid population, the frequencies are determined by the genotype frequencies, right? Allele frequencies. So if you're given this data set, how do you determine the frequency of big A and the frequency of little a from this set here? These are the genotypes. And it's pretty straightforward, right? So we know that the frequency of big A, that's what that F is going to be the frequency of big A, right? Because there's two big A's there, which is 0.2. And then plus 1/2 the frequency of big A, little a. And the reason that it's 1/2 is because they only have half of a big A allele, right? So that would be plus 0.4 divided by 2, which should be 0.2 plus zero point 0.2, which is 0.4. Right? And then the frequency of little a would be the frequency of, well, frequency of little a is 1/2 of the heterozygote because there's only one little a there, plus the frequency of the homozygous recessive because it has two little a's. So we got 0.4 divided by 2 for this one here. And then plus, so that would be 0.2 plus 0.4, which is for little a, and that gives us 0.6. And of course, we know that these have to add up to 1. So 0.6 plus 0.4 equals 100% or 1, same value. So that's how we can figure out the allele frequencies from the genotypic frequencies. All right. So if we already know the allele frequencies, we can predict the genotype frequencies. And it's not always 100% accurate because it's just a prediction. It's like I predict that you're going to flip a coin and it's going to come up 5 heads and 5 tails. That doesn't necessarily mean that it's going to be accurate every time. It's just giving you an idea. So if you're going to make a prediction, you need to use Hardy-Weinberg. If you're actually calculating with numbers that you know, then you shouldn't use Hardy-Weinberg. All right. So if we know the allele frequencies, right? If we know the frequency of P and the frequency of Q, then we can predict under the assumption of Hardy-Weinberg. And so there's lots of things in Hardy-Weinberg that we have to assume. But there's random mating, right? That means that, gentlemen can't prefer blondes. You have to randomly mate. There's no choosing a mate. It's just random. There is no selection, right? So you can't have any fitness, no mutation, no so when we say everything, no selection, is everything is a neutral polymorphism. There's no deleterious mutations. The size of the population has to be infinite. These are pretty ridiculous conditions, but it'll give you a rough estimate, right? If you need it. Hardy-Weinberg equilibrium is observed in most outcrossing, which means not inbreeding, right? They're outbreeding. Diploid, so not tetraploid. There were 2 in populations. And if a population is in Hardy-Weinberg equilibrium, and again, the conditions of this are insane, then the allele frequencies don't change from generation to generation. They're always the same. And so that means that it doesn't matter from one generation to another. The frequency of P is always going to be the same, and the frequency of Q is always going to be the same. Okay. All right, so here's the Hardy-Weinberg equation, and I just did this before, but we can do it again. So we're going to let A equal P, or so big A equal P and little a equal Q. We know that this is the equation. P is the homozygous dominant, 2PQ is heterozygous, Q squared is homozygous recessive. And it's 2 because it can be in either order. Okay, so let's do a real world population, right? What's the purpose of this? And so, let's say a couple comes to you and they're worried about their kid having cystic fibrosis, and you know nothing about their DNA. They have no DNA sequences. You can't tell anything, but you can tell them what the probability is in a population, right, of them being carriers because neither one of them have cystic fibrosis. So if you're looking at this couple, they could be, let's just use little c. So we say little c is you have cystic fibrosis, and the couple could be big C, or big C, little c, right? We don't know because they look pretty normal to us and they don't have any cystic fibrosis symptoms. So they want to know what the probability is that they're going to have a kid that has cystic fibrosis. Or in other words, what the probability, like what the problem, first of all, you have to figure out what the probability is they're carriers. You know nothing about their family history. There's no pedigrees or anything like that. You can use Heidi Weinberg to predict this. So this is how it works. You know that there's statistical values. You can go around and look at the number of live births for cystic fibrosis. Let's say there's 2,500. Uh, 1 out of 2,500 live births is an individual with cystic fibrosis. Well, you just figured out Q squared, right? Sorry. So Q squared equals 1 divided by 2,500. If we do the math, it's a very small number, 0.0004. All right. So this is Q squared. Now we want to find out Q, right? Because that's what we need. So we're going to square root this. And if we square root this, you should get 0.0004 equals 0.02. So remember, P equals 1 minus Q. So 1 minus 0.02 equals 0.98. So now we can tell the couple, okay, the probability that your 2PQ equals 2 times P, which is right here, times Q, which is right here, and that equals 0.0392 or 3.92%. So there's a 3.92% that the husband is a carrier and a 3.92% probability that the wife is also a carrier. And then you can use that to calculate, if they had a kid and they're both carriers, then they would have a 1 in 4 chance of passing that on. And so we know, how to do that math from the problem set that I gave you. But that's how we can figure out what the probability is an individual would be like a carrier for cystic fibrosis or any disease for that matter. Oops. Okay, we talked about this. Hardy Weinberg assumes infinite population size. Alleles frequencies don't change. Uh, in the real world, populations are finite. The allele frequencies change to all kinds of factors, one of which is genetic drift. And I think I'm just going to talk about genetic drift real quick, and then we'll wrap this up. So remember the let's just say that there's orange widgets out in the wild, and they like to eat purple flowered plants. So we know that the purple allele is going to be big P, and the white allele is going to be little p. All right. So what do you think is going to happen to the purple allele if the animals like to eat purple flowers? This is going to go down, right? And so if that goes down, because you know these added together have to be 100%. If that goes down, this has to go up percentage wise, right? Because we're talking about probabilities. And so I lost my train of thought. We're doing natural selection. So allele frequency varies in a population due to natural selection. Remember we talked about the pepper moss? So the black moss with the black trees got eaten, so their frequency went down. The frequency of the black allele went down. If the trees were white, the frequency of the white allele went. Sorry, the let me back up. The black moss on the black trees, the frequency of the black allele increased because they're camouflaged. The frequency of the white moth allele, the white allele, decreased in that population. And then it flipped. So we can have natural selection, right? This is what Darwin showed when he looked at the finches. He didn't really look at the turtles because he thought the turtles were from the Seychelles. And the reason that he thought that is because back in the day, sailors would take turtles and throw them in the bottom of the ship because turtles can last a long time without food or water. And then when they got hungry, they would just eat the turtles. So Darwin thought that sailors from India had released some giant tortoises from the Seychelles Islands off the coast of India in the Galapagos Islands. He didn't know that they evolved simultaneously at different places to be giant tortoises. Um, so he ignored them and he looked at the finches with their different beaks. So anyway, uh, that's natural selection, right? We're selecting it. We're selecting it. Either nature selecting it with their artificial selection. So if we're taking one dog and breeding it to another dog to make it have a flat face, right? We keep breeding it with flat face dogs until we get a flat face dog. That's artificially changing the allele frequency for flat faces versus pointy faces in dogs. So there's natural and artificial selection. They're both selective things. They put pressure on, um, we're selectively breeding things. So it's not random mating. It's not a finite population. So we get changes in allele frequencies from that. Now, genetic drift is different. So if we look at genetic drift, and let's say that we'll just pick something that doesn't matter. It's not going to change your fitness. Let's just say it's hair color. So, I hate this with it. So let's just say brown is big B and blonde is little b. Okay, so if you had two parents that were heterozygous for blonde hair and brown hair, so they're both brown haired, because this is dominant and this is recessive. Doesn't change their fitness, right? So it means they're not going to be embryonic lethal or anything. So we do the Punnett square for mom, Punnett square for dad's gametes. We can draw some tails on here just to identify dad. And then we have big B, big B. This sperm fertilizes this egg. This sperm fertilizes this egg. Big B, little b. This sperm fertilizes this egg. Big B, little b. Right? It would be little b, big B, but I just flipped it around. And then this one is little b, little b. Okay. So, what's the original frequency in the population? We have mom and we have dad. So, what's the frequency of big B? Well, that's 2 big B's out of 4 total, which is 1/2, right? We reduce that. 50%. 50% of our population is big B, and then the same for little b, right? There's 2 little b's out of 4, and so that's 1/2, and again, that's 50%, which is 0.5. All right. So now we're gonna change our population because we're gonna have a baby. So what baby is it gonna be? Well, let's say it's this baby. Okay, that baby. This is our population. This is not matings, right? So how many big B's do we have? Well, we have for big B we have 3 out of 6, which is the same as 1/2, which is 0.5, which is 50%. And little b is also 3 out of 6, which is 1/2, which is 0.5, which is 50%. Okay, so did our allele frequency change? No, it didn't. But you could have just easily had this baby. So let's say we didn't have this one, and we had that other one instead. This is just by random chance. So let's redo our values. I guess I need the eraser after all. So let's erase this. Stop doing that. Now, let's say we have this kid, the little b. So now we have our family, our happy family, mom and dad and baby, and it's a blonde baby. So what's the allele frequency? So big B equals 2 divided by 246, which is a third, and little b is 1, 2, 3, 4. 4 divided by 6, which is 2/3. Which so this is 33.3%. This is 66.6%. Hopefully you know that 0.33333 is 33.3% and 0.6666 is 66.6%. So did the allele frequency change? Well, yeah, our original allele frequency was 50% for both big B and little b. So now big B is 33%. So big B went down and little b is now 66%, so that went up. Was this done by any other way than dumb luck? And the answer is no. And so that's why we call this genetic drift. Genetic drift is just random chance of varying the population by the offspring you have. And so if we have just both heterozygotes, right, then that wouldn't change our population. There's a 50% chance that our population won't change, it won't drift, and it won't evolve. So just, I'm going to introduce the dreaded E word. So evolution is change in allele frequency. So it doesn't matter if the frequency change is due to genetic drift or selection, right? Either natural or artificial selection. If it changes, then it evolved. So there's a 50% chance if you have a kid that it's not going to change. But there's also a 50% chance that it will, right? And so we're gonna talk about this in the next lecture because I'm gonna wrap this up. It's like almost an hour and 15 minutes. But um, genetic drift, just random chance, has a tendency to make a population homozygous. And I'm just gonna say that, and then I'll show you why that is. All right. So that's kind of my cliffhanger there. I'm sure you're all at the edge of your seats. We'll pick this up. Um, I'll do another lecture probably tomorrow or the next day, and then I'll post it. So anyway, if you have any questions, email me, um, or you can send me an email at Canvas or send it to my email address, or come to office hours. Uh, Otherwise, I'll see you next time.